Hi everybody, welcome to the webinar today for using flow cytometry bead-based assays to characterize, calibrate, and produce high quality and reproducible results. The webinar today is a joint effort between Spherotech and Novo Software. My name is Sean Burke from Novo Software, and I'm joined on the call today by Michael Collis from Spherotech. So welcome to the call, everybody. Welcome to the call, Michael, and thanks everybody for joining us today. So to get started on the webinar, the teams at Novo Software and Spherotech are really pleased to be working together today on this webinar. Essentially, we've started collaborating so that we can kind of help researchers more easily move from their beads to results. By using the high quality beads that are coming from Spherotech and then the easy to use data analysis and reporting within FCS Express. So the first half of today's webinar is going to focus on the capabilities for alignment, calibration, and getting quantitative results from Spherotech's line of beads. The team at Novo Software are then going to follow up, and we're going to actually look at some of this real bead data in FCS Express and show you how to produce some reports for reporting alignments, reporting linearity, reporting calibration, all of which are going to be done directly in FCS Express without having to use external softwares like Excel or Prism or macros and things like that. And we're also going to come back at the end and talk about how to use the channel calibration tool in FCS Express to automatically convert raw data values into MESF values. Now, after the webinar today, the teams at Spherotech and Novo Software are going to try and make some of these templates that we're working with today available for researchers in general. So make sure to keep an eye out in your email. When we send you a message, you'll be able to access some of these templates. If your instruments and your configurations are similar, it'll be a good place to start off. If they're not, both teams are here to help support you. So with that, I'd like to introduce Michael Collis. Michael, we got you there today?
Yeah, thank you, Sean. Thank you so
Great.
much. And thank you, everyone.
Let me make you the presenter here. And you should be able to show your screen and looks good. And we'll let you take it away from there.
Fabulous. And thank you, Sean. And thank you, everyone here today for joining us for this webinar. We're really excited to be talking about beads and some technology to help you make your lab and your QC a lot easier as you move forward and even potentially help quantify your results and analysis. And I also like to thank on the team here from Novo, Raphael, Juan, and Sean, who helped us out, and my mentor, Brian, at here at Spherotech, that many of you may know. So on the agenda, we're just going to start off with some introductions, goals, and objectives, a little bit about us and what Particles can do for you, but really the meat and potatoes of why we're here today is for this portfolio of capabilities: alignment, calibration, quantitative intensity identification for MEF values, and determining the background noise and low-end sensitivity of your various instruments. And these work across all instruments. And as Sean from Novo had mentioned, we are working on making instrument-specific templates and macros so that they can work for not only the individual instruments in your core, but maybe you have multiple at your disposal and you want to normalize them or use them together. So where is Spherotech? Spherotech's just north of Chicago, right? We've been here for a little over, come up for 30 years now, 1992. We're kind of a small outfit, but we always like to talk about how flexible we are, and so we're able to meet you in very unique capabilities, still being such a small and agile organization. We are ISO 9001 designated. This is actually our 2015 certificate as opposed to 2008 rather, registered. But just in case, all that kind of good stuff with quality and approval. But really, what are we talking about today is you all in the field, right? We're talking about researchers, virologists, geneticists, proteomics, and all kinds of research, and down to just flow cores. How can you meet the needs of your university or your organization so that they can push science to the next level, as well as at universities, hospitals, and large organizations. But microparticles. So why do we care about beads, or what beads are we talking about? Sometimes you hear about spheres, beads, particles. And we're talking about latex, typically polystyrene. We also do offer polymethyl methacrylate, silica. We do have some gold nanoparticles, which are very cool, and some new capabilities therein. But this is really our bread and butter, and one of my favorite SEM photos here of some of our nanoparticles on hair. Rather 100 micron hair and 3 micron nanoparticles. We won't share whose hair that is, of course. But really, we're talking about optical comparatives. That's what these beads mean to you. They are rulers for that cellular and subcellular world, right? Previously, maybe you do it today, maybe in another life, you had to grow and culture and keep a set of cells inside of your lab that you would then stain potentially as controls, keep as blanks and positive controls, as well as potentially actually creating calibration curves where you had to titrate the amount of dye that you put onto these cells. And of course, you would do all of this hard work, or your techs would do all of this hard work for weeks, and then the cells would no longer be viable. You would have to replace them, and you'd have to reset up all of your calibration curves based on those new cells, that new population. Right? So if you have these longitudinal studies over multiple years, or you're studying multiple generations of that same bacteria over time, or that virus over time, how are you going to keep your controls the same? And this has always been a big challenge. And that's one of the solutions that we provide with these wonderful microparticles. And today I really want to talk about not just individually fluorescent particles, but rainbow calibration particles that we're going to be talking about here, these quantitative. I know I'm jumping around a little bit, but so let's talk about value add just for a half second before we get to that quality control. And this is kind of the cool stuff that we can offer you. As I was mentioning, many of you are researchers, or maybe you're conjugating various things to your beads. You're doing antibody assays, you're doing PCR, you could be doing all kinds of purification and quantification. We can offer custom work with this, and we're actually also, we haven't talked about it quite yet, but we are offering new click chemistry particles, if any of you want to ask or inquire about that in the near future. Azide and DBCO, copper-free click chemistry, that's really precise and really nice. We've always offered particles for EDAC chemistry, like carboxyl and amine, for standard conjugations of proteins and or oligos. But now we're looking into the click, which could be a really cool cycloaddition Huixen, if any of you are familiar with the chemistry. But let's talk more about these product lines, these calibration particles we have highlighted here, and these rainbow QC kits, right? We also offer absolute counting, compensation, multiplex. We may work with de novo in the near future on webinars regarding these other capabilities and how both of us can kind of help some of these turnkey solutions that we'll talk about, or some of these easy to use, as much fun as it is working with the beads, we have these templates that we had made in Excel that can sometimes be clunky. The amazing thing about de novo is you can take this raw data that you've produced with these highly technologically advanced particles and just dump in your FCS file, and immediately you have your SOPs that'll populate with all kinds of fun things. I mean, Sean's going to touch on it later, but it's amazing the ability that you have when you're given strong and capable tools. And that's what we're here to give you today. And we also do offer magnetic separators for various things outside of just particles. So here, these are our particle types, right? So at the top, we have alignment. We have rainbow, ultra rainbow, and supra rainbow. Now, rainbow are gonna work off of your regular, if you have a 488 laser, your 561s, your blue, your yellow, and your 633, your red laser, those standards, or if you even have the violet laser, these are all gonna, the 405, these are all gonna work really well with the rainbows. Ultra rainbows have some UV capabilities, as well as they were developed about 10 or 15 years after. They're a little bit cleaner, and the supras offer kind of an interesting mix of capabilities between the two and a few really nice alignment particles. So those examples here are RFP for the rainbow, UR for the ultra rainbow, and SR for the supra, right? These are all single peak fluorescent precision particles. And when I say single peak, they do have all the colors of the rainbow inside of one particle. right? So we have here an, kind of a fuzzy image, but you can see the concert of all of the different colors of dyes that we would have. Inside of a rainbow particle, we're putting all of those together so that regardless of the excitation line that we use, whether we use a UV coming out here, or whether we use a, this is off the blue laser, this would be off of the violet laser here, and this off of the red laser. So these are all clearly the same beads in the same clustered population, but based on the excitation, you'll have different emission, which is very, very powerful. It means one bead for every channel inside of your flow cytometer. The calibration particles take this one step further by using multiple intensities. So you'll actually get a series of populations, one slightly brighter than the last, or rather exponentially brighter than the last, so that it fills up the entire dynamic range of your instrument, whether it be a 10 to the 5th or a 10 to the 7th, whether they be BD, Cytech, Becton Decker, all kinds of, we have Coulter, all kinds of instruments out in the field there nowadays. We consult on many of them and have a number in-house. Likewise, we have these multi-flow, these multi-peak particles. We have the traditional rainbow 8-peak, 6-peak. Euroflow, for any of you that are in the Eurozone or do follow the council, we have a specific Euroflow council determined MFI variance ranges for these Euroflow particles, but they come in the exact same kind of capabilities as the previous, as well as NIST. And a few of you who may be interested in some of these seminal vesicles or some of the nano world, not only do we have these rainbow calibration particles in 4-peak, we have a few alignment particles in this range, and some of our R&D over the last few months and into the next few are also going to be focused on this kind of nano world. So again, like I said, one bead potentially to rule them all. So we're talking about all of our channels at once, right? This is a standard breakdown of what we would consider a flow cytometer, right? We have our laser line coming, our fluidic system, and then off of this series of dichroic mirrors, we have our PMTs, or avalanche photodiodes, depending upon your system, right? And given these different bandpass filters, you'll be able to capture different data for each one. However, with these rainbows, of course, as many of you know, it works in all channels. So we're able to calibrate across all of these channels simultaneously. And we're going to get into that momentarily. And also just make sure, kind of do a check-in, make sure your instrument's working well. So particle preparation. Sometimes this is kind of glanced over, glossed over, but it's good to take a minute. I know it's very simple, but we store these particles typically at standard temperature and pressure, but preferably, whenever possible, refrigerated, just to reduce any potential microbials or just for the longevity of the particles. We don't typically want to freeze them, right? So these particles, being mostly 3 micron, are going to settle over time inside of the bottle. So whenever you pick up the bottle, you want to give it a shake, you want to agitate it, you might want to vortex it, right? You're going to dilute just a drop or two, up to about 2 mLs, right? If every drop is about 25, right, microliters, is that about right? You want to do at least an order or 2 magnitude dilution. They start off at about 1e to the 7th, anywhere between 1e to the 5th and 1e to the 7th is a good final concentration. And when you're getting our less precise particles that may come in higher percentages or higher concentrations, it's always good to dilute to about this level into a flow cytometer, not only so that your event rates are reasonable, but so that you don't end up cleaning it. You know, if you put in very, very concentrated particles, they're much easier to visualize, but then potentially the next users or the flow core staff are going to have to spend quite a bit of time cleaning that instrument. So we want to make sure that our particles are present, but not too concentrated. And likewise, we have both of these examples, our rainbow calibration particles, 8 peaks coming in a single unit, and our ultra rainbow calibration particle kit, where you get 1 fluorescent population, with rather 5, and 1 blank population. Okay, so these are 2 examples of some flow cytometers we have here at Spherotech. This is our Cytoflex on the right and our Fortessa on the left. The previous image that you had seen was from our Nikon Eclipse 2 spectral microscope. And we have some SEM and a few other instruments in-house. But these are our two primary workhorses when it comes to cytometry. And so we're going to start with alignment. We're going to start with the single peak particles today. And just as an example, I have some front scatter versus side scatter here. I certainly could have made the dirty a lot dirtier. You know, this entire frame could potentially just be eaten up. We all know if you put in a way too dense of a sample. But when you see a population like this, I just wanted to include it as perfectly or almost rather diagonally linearly from your main population. There's a good chance this is a doublet as opposed to just debris, right? So if you're only getting like 60%, 70% singlets here, this might well be a doublet population that we need to vortex, right? We were talking about agitating the particles. A lot of times with a bit of agitation, this will come down. Now, if it's not on this kind of line, on this linearity here, there's less of a chance that it's a doublet, but just as kind of an example. So alignment is to make sure that our avalanche photodiodes, not ADP, we do want to get paid. Everybody loves ADP, but APD, avalanche photodiodes, and PMTs, right, photomultiplier tubes, we want to make sure they're functioning optimally, right? And what does that really mean? Well, it comes down to MFIs, intensity, and the CVs, or the width of those peaks, right? So like I said, single peak ultra rainbows are what I'm running here, the ultra rainbow fluorescent particle 30-2. I like to put my peak at 10 to the 5th, right? Some people will argue to put it at 10 to the 4th so you can see the brightness below and above, right? But my standard, and like I said, you want to set your own SOPs for your labs, and Novo has this wonderful software where you can actually program in your SOPs so that you know if you're a quality assurance specialist that your QC staff or that your end users are always doing the exact precise thing. There are these great SOPs in Novo that will help you. But we typically put this at 100, and then read pretty boring, but it's always good to keep this, the voltages that you set for the day, it's always good to have this on hand or in your records. And these alignment particles day to day might not seem terribly interesting, but what we're looking for is these very slight jumps that will happen left and right, or widths, right? In order to determine, and then over 30 days, I collect this same piece of data. And I only did it for 15, and I did it across, you know, a number of channels here. And you want to set acceptable ranges. And the only way to determine these acceptable ranges for your instrument is to measure it longitudinally. So once you have 15, 20, 30 days worth of data, you can say, if this varies beyond what I'm seeing here, if this varies beyond what I'm seeing here in front scatter or side scatter, I could see that as unacceptable, or the point at which I need to call my instrument manufacturer, right? Double check to make sure that my lasers are in alignment and that my in alignment not just with the sample or the fluidic system, but also with my my filters, right? And you'll be able to tell this from the CV, or the width of this peak, as well as the intensity of this peak. You want it to be very consistent, right, over time. And you'll see that the means are going to vary more than the CVs, of course, and the scale, you know, as you can see, my scale jumps. So this variance looks a lot larger than it would be if this scale went all the way down to 0. But I just wanted to create a couple of quick and easy plots to say, okay, if this value drops below 9,000 or above 9,800, that's not typical for this FTSE, right? For this FTSE. And likewise for this per CP, it's only about a variance of 10,000 over you know, a 10 to the 7th range, but you want to make sure that it falls consistently. So this is really the power of the alignment particle, is determining your day-to-day health of your instrument, making sure that it can see something very precise. Next, so this is more about precision. Next, we're going to focus on accuracy, and that is its ability to tell or differentiate between different brightnesses, right? So this is our 8-peak product. Many of you may be familiar with this. This has been the flagship particle for flow cytometry for almost 30 years now. We gate the 8 peaks. I typically, we'll show a different example, but this is the standard where you would do a horizontal fence gating across the FTSE channel, across the entire dynamic range, and then allow that to show up in each channel. Some SOPs will tell you to gate every single channel individually. And I actually prefer doing this. I like crossing like an APC FTSE or a PEN FTSE and actually doing a full population gate around the peaks. And I can also then see how it goes across my dynamic range. And we'll get to why this box is here a little bit later. But this is some BD data from my Fortessa as a quick example. And again, I'm saving those voltages, because anything that we're doing with our flow cytometer is only meaningful if we maintain voltages across different runs. You know, if I run my ultra calibration particles here and I determine the linearity of my instrument, but I change the voltage before I put in my experimental value, when I'm going to determine that unknown brightness, it's no longer meaningful, it's no longer significant, right? So we need to maintain. You can change your front and side scatter. You want to maintain these fluorescent voltages. Okay, so here are some examples here, and I have run these various data. What you want to do is you want to grab the mean and the CV, and really the mean for every single channel. The CVs, you'll notice, will decrease. 21, 8, right, 6, 5, oh, down to 5. Oh, I like that peak, right? I want my CVs, oh, 4.4, I really like that peak. Okay, enough about numbers. But so we want to see these intensity values increase. And we're going to actually, you know, some people will mention, oh, this is an exponential curve. Do we have to apply an exponential log in order to exponential formula in order to determine where we're going? And what I usually respond with is that we're actually going to do the inverse. We're going to use a log in order to linearize it. The first log that we apply, you don't necessarily need to think too much about this, but the first log that we apply is based on the actual number of bins per bit, the electronic capabilities of your photomultiplier tube or your avalanche photodiode. So we account for the number of bits. Let's say you have an 18-bit system versus a 16 or 24-bit. We first account for that, and then secondly, we account for the log across this dynamic range and make it a relative value. Now, these, thankfully, is all done by our macros. You can go to our macros home, let's write this technical tab on our website, and this will open up our templates home. Now, these templates are not as wonderful and easy to use and malleable as the ones at de Novo are, and that's why we're collaborating with them, being the experts in that capability. But these will provide for you pretty classic Excel templates. And like Sean had said, you know, we're going to be integrating a lot of the ones that we have custom built for certain instruments. So if you have a Cytoflex, or you say I have an Aurora, or if you have a Quantion Novasight, and you say I need the template to be foolproof, I want any new employee or any day-to-day user to be able to use this template, right, without any sort of aid. That's where we're going to get them with de Novo. But just for the nuts and bolts, we have them here. And they're all lot specific, right? Because each lot is slightly different. Likewise, I have these Spherotechnical notes. If you have follow-up questions, we can talk about them. But this one, 9, STN9, if you're going to put a star next to any of them, measuring MEF with a flow cytometer using the rainbow calibration particles, this is the one to highlight. Okay, so this is what the macros look like. But what we're going to do is we're looking for this linearity. We want a very, very high confidence on this linearity. So like I said before, so you run your particles, you gate your particles, you get that mean value, right? You get that mean value. And we're going to convert this mean value inside of this macros. Depending upon your instrument, you would put in, oh, I have a 10 to the 5th channel, so I'm going to need to convert it with this table. Once I get my converted, my 256 values, plug them in here, and it will spit out a linearity for me. This one, of course, is set to 100. And likewise, if you call me up, you ask me, hey, where do I put the numbers? Where do I need to go next? We can always help you with this. But like I said, de Novo is going to streamline this at least 2 steps. But as you can see, we can also normalize different instruments. And we're going to get into this cross-calibration table in a second. Okay, so back to my data, right? Let's jump into the cross-calibration table now. So you would regularly put in your 6 or 8 data points. You will get this converted value. And you're looking in here for an R squared. If you want to write down a good R squared, you're going to want at least like a 99.2. I would shoot for a 99.5, right? A 0.05% confidence level usually makes me pretty happy. We like to beat 99.7 in most channels. There's only 1 or 2 channels that should give us any trouble. If you see any 98s, now, those are better than some of my grades from college, but if you see any 98s, you want to call somebody. You shouldn't be seeing that kind of linearity inside of your system. These are highly correlated values. Okay, so you're looking for a linearity of 999. And for a lot of you, that's going to be all you need to do. You plug in your mean values here. You plug in your relative values here. As long as this is over 999 or 995, boom, the cytometer is ready to go today. For some of us, we might say, well, what if now I want to determine an unknown sample? I want to determine the brightness of an unknown sample. And in fact, I have a juvenile and I have a mature cell line. And I want to know if the expression of a specific surface marker is more prevalent on the mature or the juvenile, or can I say something significant therein? Yes, that's the power of these beads at the end of the day, is being able to do unknown population brightness determination. Now, we're not going to necessarily be using an absolute value, because these are all correlative values. But if you know the concentration of your reactant, the amount of which bound, you can say something significant then about any ratio that you make between it and a secondary population. The juveniles have 1/3 the amount of expression on average, significantly over, let's say, 10,000 events, right? That's exactly what we're looking to do here. So once we have our linearity 999, let's say, on these exact same settings, I'm not going to change any settings. I run some unknown population, right? And this unknown population seems to be low in APC, but high in FITC. And it also shows up in my PE channels. So let's say I want to determine how bright it actually is. Well, it looks kind of like this peak, right? But we don't want to know kind of. We want to be able to say with confidence, this has a specific MEF value. So as I said before, and this is just the macros broken down, right? If we go back here, this is the main table. I'm going to use this table, and the graph. That's all we're going to see here. So this little table here is me putting in my mean value. Mean values go in, and I call them 256, but these relative values come out, and I put them in my main table. Now these in red are my validated values. We've given you those. Spherotech gave you those values. But if you put in your channel numbers, you will get your calculated MEF values. And look at that. I got a great R squared value, 999, right? It's pretty low residuals. So now I've essentially got the table set up. Now I'm going to put in my unknown. Remember, I got my unknown here. Well, that had a mean. I'm going to pump it just in the bottom here. I'm just going to put it right here, pop out my relative value, and then this cross-calibration table. Remember I told you back here, we've only been talking about the left side of the table, right here in the middle, cross-calibration, right? That's going to be from my unknown samples. So I can put in, oh, this is the name of my sample. This was my relative channel number. As long as this is set up, the macros are made to tell you, oh, based on this linearity, if you have this relative channel value, this is your MEF. And then visually, I saw it was around that second to brightest peak. Well, does it have a similar MEF? Yes. So that's good. Probably means the macros are all talking to one another. This is some of the power that we're talking about. You know, if you do this with 15 or 20 samples, if you do this with a longitudinal study, not only that, but the beads stay the same. Unlike your viability of cells, you can say, 5 years later, the brightness of the beads is the same, or at the very least, it's validated. And if you don't feel comfortable enough with our validation of MEF, which is highly vetted, and I do have the literature, but NIST, I do have NIST particles. They're slightly more expensive, of course, but only slightly. And they come with the confidence that NIST has given it. And I also have some literature that NIST has written themselves comparing our MEF values to their ERF, right? We say molecules of equivalent fluorophore. They say equivalent reference fluorochrome. It's very, very similar. And then likewise, kind of as one of my last points, coming into, we've done alignment, done some calibration. Sure, we're going to do questions, but low-end sensitivity, right, and background noise. We want to find these values, and it's as opposed to before where we had this linearity of multiple brightnesses and these very, very bright values. We're still going to need that calibration particle, but what we're doing is we're looking down here. We're going to put in some dim peaks to see if this linearity, or rather the vision capabilities, is following the same trend in this very, very low end area of our flow cytometer, right? We want to see essentially it's like asking how good are the eyes of the person you trust, right? You want to make sure that they have good glasses, or at least you have a corrective value to determine what they're seeing, right? So we can use the blank from the first series as a first dim population. You're going to need a second dim population. Now, you could probably use a cell, you could use a single fluor particle. We offer some FL, or fluorescence low intensity particles. As well as you can always grab like a single peak. A lot of our multi-peak products you can order as just the second peak or just the third peak if it's an 8-peak particle. And inside of the macros, we also offer these, but it's a fairly straightforward calculation. But what you would do is you would run the 8-peak particles or the 6-peak particles, and then you run your 2 dim particle populations, and then essentially you're just doing you're squaring and square rooting these values in order to determine the efficiency, the efficiency and the background noise. So you could say that what the minimum possible is. And the other reason why this is really powerful or really good is because a lot of the time you'll notice if you play with the voltages, your first peak might go to a negative mean value. And since we're doing logs, you can't do the log of a negative value. So there's an area with which you can't say something with confidence inside of your instrument. So essentially what this does is it creates a linearity or a regression line into that unknown area. So maybe this has a slope of 1 or 2, whereas down here you have a slope of 0.1, 0.1, right? Something like that, where the sensitivity in the first third of your cytometer's window
sales
is
line
weaker
2
than the higher sections. But yeah, I just wanted to finish up by holding up a slide of our top products. But I think we're coming to about where I need to be calling it. But I really have appreciated your time here today. And I'm probably going to pass it back to Sean. We're really looking forward to any questions or capabilities herein. And yeah, thank you. Thank you so much.
Great, thanks, Michael. And that was a great overview of the beads and some of the current workflows that Spherotech is offering. And again, what Michael highlighted there, a lot of the Excel templates and macros that are there is pretty much where we're going to pick up in FCS Express now, because by using FCS Express and using the integrated spreadsheets and graphing functionality, essentially what you can do is, instead of having to move between data analysis and Excel macros and then punching in numbers and trying to get out a kind of static result, we can start working with all that data and get information in real time in FCS Express. So literally, as you move a gate, you can get MEF values, right? As you move a gate, you can see how your CVs update across multiple longitudinal studies and things like that. So in this portion of the webinar, I'm going to pick up and we're going to talk about some of the data analysis from these beads in FCS Express. Before we do so, I just want to give you a little bit of background. I know not everybody here is using FCS Express yet. That's okay. We know that you're going to come over to us very soon if you're not. But keep in mind, we've been doing this for over 20 years now at Novo Software, right? FCS Express has been around a really long time with global support and applications teams for unrivaled support. And really what that means is, if you have a question about anything really, if it's relation to the beads, we're happy to help you out. out, but shoot us an email at support@denovosoftware.com, and we'll be able to get you up and running, either with new templates or whatever templates are offered, so you can get going. Now, what you're going to see when we use FCS Express today is that it's a fully integrated analysis, statistics, graphing, and reporting tool, all meant to get you results. And because the entire software package is kind of a Microsoft Office inspired design and interface, it makes learning FCS Express very easy. So if you've ever used PowerPoint, if you've ever used Excel, you're going to have that same kind of functionality in FCS Express, but directly linked to your data and gates. And any of the publication kind of ready or lab notebook ready results that you're going to get are going to come out in super high quality, super high resolution. And again, you're not going to be stuck using multiple software tools just to get a result. So we're going to look at 3 use cases in FCS Express. So one is looking at some of the alignment and linearity, which you kind of saw in the Excel macros coming from the beads that Michael showed off. We're going to take a look at a kind of Levi-Jennings style analysis. So looking at a longitudinal study to make sure CVs and MFI values are staying the same and staying within range across multiple days. And then we're going to be coming back around and talking about calibration. So being able to go back and say use a known set of MEF or MESF standards and then calibrate your raw data to that, and not just taking a population level number of MFI, plugging that into a macro, but actually getting those results as you move gates, as you look at data. So the results that you're reporting in the software where you're performing analysis are already calibrated for you as you're looking at them. So this is all done through integrated spreadsheets and graphing. And again, what I'm going to do is switch over to FCS Express here. And don't get fooled, this is FCS Express. I know it looks exactly like Microsoft PowerPoint. That is, again, shamelessly by design. We know everybody here knows how to use PowerPoint, right? So when we look at this layout in FCS Express, you can see here that there's nothing here yet, right? It just kind of looks like a bunch of empty plots and information. But if we have a template set up, either one that you pull down from our website, hopefully from the Spherotech website, or even one that you can create your own, essentially the way that we populate this template is we drag and drop one of our bead-based data sets, and this is a 6-peak ultra rainbow calibration particle. It was run on a BD instrument, and you can see that these plots populate, right? So everything is done for you here. That's one of the really nice things about using layouts in FCS Express as a template, is that essentially all of your analysis is already kind of set up and done for you. Now, in this template, we have a gate set up to kind of identify where the beads are. We have some of these gates set up here to identify, again, kind of further where those beads are, and also some plots to help, support the visualization. So looking at, different overlays of backgated populations and histograms, which is a very effective way of doing this. The other thing we can do in FCS Express is use multiple pages and tabs to help organize our data. So with this particular instrument, it's a 4-laser system. There's a violet, blue, yellow, green, and red laser. So what we've chosen to do is for every single laser line, or every single laser, show all of the different parameters that are being acquired on that laser. So in this case, we have BV421 through BV786. On our blue page, we have all of our parameters outlined for things coming off of the blue laser, and again, the yellow, green, and the red. So again, just using these page tabs is a great way to organize this data. Now, one thing that you're going to notice is that on every page, we have multiple kind of analysis spreadsheets here. And if you remember back to what Michael showed you, there's some Excel macros and Excel programs that you can download from the Spherotech website. Well, these are effectively recreated here in FCS Express. So we're pulling an MFI value. We're doing the conversions that are set up in those templates for you in the Spherotech macro. macros. We have the MEFL values that are a lot specific in here. And again, a whole bunch of other calculations that are here to arrive at the slope, the intercept, and the R-squared values. And again, this is being done for every parameter. Now, the really nice thing about this is that these MFI values are just not, copy and pasted tables into a macro. They actually are linked and represent the data that's visualized on this page. So for instance, if I highlight this, Excel cell here, this spreadsheet cell, if I move marker 2, what you're going to see is that cell in that spreadsheet will update. And of course, all of the calculations that that's based off of, slopes, intercepts, R-squareds, that information updates immediately as well. So again, instead of having to copy and paste and move to external softwares to grab this information, you're getting it reported all within FCS Express. Now, to take that a little bit further, I know that many folks are used to looking at the linearity curves or the linearity scores, and we can achieve that in FCS Express by using our scatter plots and our scatter with regression plots. So what we get is for every single channel, we can look at the R-squareds, we can look at the slopes, we can look at this data, figure out if this is good, if it's bad. Again, most of these are 0.999 or more, which again, it's meant to be very linear. Our instrument is performing well. But additionally, when we're ready to, set gates or we need to review this data, as we move gates in FCS Express, we're not going to have to re-export that information out to an Excel kind of macro. You can see here that as I move this gate around, even if I grab some of this other stuff that might not be so ideal, all of our regression plots are updating. And again, they're not updating a big deal because our instrument is very well aligned and calibrated. It's very linear in its nature, so we're not getting big differences here. But I think it's important to point out that you get this live updating information. Now, when it comes to reporting this, you can very simply come up to the batch actions. You can click run. If you have any sort of reporting already set up, like exporting the PowerPoint or the PDF, the high resolution images, whatever it is that you want to export to. And FCS Express is going to collate all of the information across all of these pages. It's going to put that into a PowerPoint presentation for you. It'll put it into a PDF document if you want. And you can see here that PowerPoint has launched up. We have all of our information, we have all of our reports, we have all of our regressions. And again, these are super high quality graphics in FCS Express, and they're what's called vector graphics. So you can move and resize and grab and pick and choose what you need for what you need to report, whether it be a lab notebook or something for future record keeping. Now, in this particular assay, this is the 6-peak ultra rainbow calibration particles, but keep in mind this can be scaled, right, through use of additional templates, setting up your own templates, depending on how many parameters you have available on your instrument. In this case, I loaded up some 8-peak data from the rainbow calibration particles, and you can see it works the same exact way. Right, you load in your data into the data list. You have a few pages set up to help kind of calibrate your data. You can have pages at the end set up to look at some of the regressions and the regression statistics, and you're kind of off and running. All right, so working with these linearity type assays, it's very simple and easy in FCS Express, and it can all be achieved directly in the same software without having to move between additional tools and use macros and things like that. Now, when it comes to looking at this data and these characterizations longitudinally, over many days, well, again, FCS Express can be used to measure this information. So what we have here is 10 data files. These are data files that were acquired between September 28th and October 9th of this year. And again, what we're trying to do is measure CVs and MFIs across time to make sure that our instrument is staying calibrated. So again, we have all of our values being reported here for forward scatter, side scatter CVs, FITC MFIs, FITC CVs, and really across all the different channels of interest. interest. But again, once we have these spreadsheets populated, and I'll show you how to do that in just a moment, you can generate graphs, right? These Levi Jennings styled graphs. I should say it's not a true Levi Jennings plot that's coming very soon in FCS Express, but you can see that we can very easily look across our side scatter, our forward scatter, all of our different parameters and see what these graphs look like across time. Now, additionally, when these graphs, it's a lot more kind of obvious now when I move gates around how these graphs are updating for us. But we can go in and we can see, all right, well, how does our beads gating, impact the results of these CVs, right? We can go in, we can look at gates for individual data files. We can go in and make those changes and get that reporting immediately in real time without having to move between different software packages in Excel. Now, all of that is based off of this spreadsheet that I have on the first page here, right, where I have all of our CVs reported and things like that. And you're going to notice that in this spreadsheet, we have some cells highlighted, there's different colors, there's different things going on. And all of this is done using the tools, our spreadsheet layout tools, just like you'd have in Excel. You can change fonts and formatting, but importantly, you can apply conditional formatting, which allows you to set ranges and allows you to say, if a cell or a value falls above 1 or 2 standard deviations of a certain range, apply a color. If it falls 1 or 2 standard deviations below a certain range, apply a different color. Now that's what we've done here. Anything with pink text is above 1 standard deviation. Anything in red is below 1 standard deviation. And we just highlighted this slightly a different way by highlighting the background of these cells. And what you're going to find is that as gates move, as data changes, that conditional formatting will update. Now, what that's going to do for you is give you some really nice quality control at a glance in this table. So instead of having to go back to my CV graphs and see, all right, well, obviously between these few days, something happened here, right? We can actually go through and we can look in our spreadsheet and we can see which data files are the trouble, which ones are the problem. And then we can do things like navigating through our different controls, navigating through these different data files. And we can see, right, there was obviously some sort of change in the scatter here when we're looking at one day versus another. And then we can move that gate. We can shift it for one specific sample. We can shift it for all of them as a group and kind of evaluate what's going on with that data. Now you might be wondering, all right, well, how do I create these spreadsheets, right? This looks pretty cool. It looks like something that would be useful. Well, it's not hard. It's very intuitive in FCS Express. If I want to get data from my data list into the spreadsheet, I can grab a few different data files. In this case, I'll choose the last 3 days here in October. I can drag it into that spreadsheet and say, give me a statistic. Now I tell FCS Express, what statistic do I want? Let's grab forward scatter, side scatter, B525, B610. Let's choose information from the beads gate and let's put in the CV, okay? So when I click OK, that information across the 3 data files, across the 4 different parameters, is populated into the spreadsheet based on where the beads gate falls on each one of those data files. And you can see here, here's my 294, 3.89, 2.64, right? That's how I got that data into the spreadsheet. And just like formatting in Excel, I formatted the cells there to make them look a little bit nicer, to get them into a table. But because you're dragging and dropping that information that's based off of a gate into the spreadsheet, as you move a gate, that spreadsheet value updates in real time. And essentially that's how we're populating these spreadsheets, how we're populating tables, how we're populating graphs in FCS Express for really all of the things that you're doing. So again, creating your own templates is very easy, but again, hopefully we'll have a lot of these templates for you to use going forward. Now, the last kind of experiment example I want to show you a little bit about today is working with channel calibration. And when I talk about channel calibration, what I'm talking about is trying to take a known set of MESF or MEF values from a bead-based data set calculating what that kind of linear regression looks like. So I can take that and apply it back to an unknown sample, or apply it back to my immunophenotyping experiment for that day. So that way, when I report GFP or FITC or PE for any of my markers, I get an empirical result that's an actual MESF value rather than an arbitrary unit that we normally get from a flow cytometer. So the place that we start for this, and we'll kind of walk you through the process, is I get some data into FCS Express. And again, I'm going to use this 6-peak ultra rainbow calibration particle. And this is the same one that we use for linearity, right? It's the same thing that we used for calibration earlier. But because we have known MEF values with these Spherotech bead lots, we can use that to our advantage to kind of calculate a regression curve and get that applied back to some other data. Now, the template here looks almost exactly the same as the linearity ones, right? We have pages for different lasers set up. My spreadsheets are set up a little bit differently. My histograms look a little bit different. But really, what we're interested in doing here is generating a histogram for any of the markers or any of the channels that we want to do a calibration on. So in this case, I want to calibrate BV421, FITC, PE, and APC, and eventually get those calibration regressions back to some sample data. So we have a tool here in FCS Express. It's in the Tools tab, and it's called Channel Calibration. And you can see that when I open this up, there's already a series of kind of equations for different parameters that I have set up. And it's actually evaluating these 4 that I have open right now. So BV421, FITC, PE, and APC. And the way that you set these things up is you would add one, or if I can pick one, I can just edit it right now, is you choose a parameter to calibrate. You choose a histogram in which we're going to generate or look at the MFI values. So in this case, we're going to look at this particular histogram for BV421 that we have inserted. And then it asks you, how many peaks do you want to use? How many beads do you have, right? And then this is where we're going to enter in the known values provided from the Spherotech lots, right? Now, if I'm using this as a template and I've already entered these values in here at some point in the past, they're going to be retained. Just like I just opened this layout, they're already in there, they're already accessible. So as long as you're using the same lot, all you have to do is kind of switch out, the bead data file that you ran that day. So when you have this set up, that's how we calculate the curve in FCS Express. Here's, our Y values, say, our X values are, say, coming from these markers here in the histogram. And that allows FCS Express to create an equation for that particular set of markers and beads. And then what we would do is just click Save that equation. So FCS Express will save this as what's called a .cal file, and this is just a series of equations that we can use somewhere else, when we actually want to analyze some data. And what that's going to look like is I'm going to open up another layout here where we're actually going to perform some analysis. And imagine again, this is your sample, right? This is your immunophenotyping for the day. This is your known standard. This is where you want to perform an analysis and you want to have this data calibrated to some known MESF value. So again, you can see here, these are not the calibration beads, right? These are just a kind of different known standard, or these could be your cells, your immunophenotyping today. But when we look at the different pages here for FITC, PE, APC, BV421, I have a few different graphs set up, and I have in each one a spreadsheet showing us our not calibrated, which is our plot at the top, and our calibrated, which is our plot at the bottom. And the calibration hasn't been applied yet, right? So that's why they look the same, and that's why our bar graphs look the same, right? But what we do is we can now come into that channel calibration tool, and we can say, well, let's load up that channel calibration, or that series of equations that we had set up earlier, which is right here. We're going to open that up, and lo and behold, the series of equations appears in FCS Express. So I can simply just mark these as active. If I want to use these on the plots, I mark them as active. And what you're going to see when I click OK is that this plot on the bottom, the axis is going to change. The MFI values being reported in the spreadsheet are going to change. The bar graphs showing not calibrated versus calibrated MFIs are going to change. So again, keep an eye on that. I'm going to click OK. And you can see everything shifted. right? So 10 to the 1 all of a sudden is up here a little bit, right? And you can see that our calibrated sample using our series of equations, the MF5 value is now 503. And that's a value based off of the MEF regressions from known standards that you've used. And again, you can see here that we're comparing that just very quickly in a bar chart in FCS Express. So of course, these can go both ways. Sometimes the MFIs get higher, sometimes they get lower. It depends on the instruments and the beads. But in this case, right, that value shifted. And this is our actual plot. This is our actual data. So all of our values that we'd be using, say if this was an immunophenotyping experiment, are now automatically calibrated. I don't have to copy and paste the values into an Excel spreadsheet or a macro from a population level. As I move gates, as I change things, everything is being reported in as MESF values that are based on known standards and controls, right? And again, right, you can see the live updating gating. This histogram marker kind of fell off the chart a little bit there, but that calibrated value is updating, based off of where I put that marker. And if that was on live cells or, whatever you were gating downstream, it would update automatically as well. So again, I think this is all pretty simple stuff to do, right? Creating the templates, isn't very difficult. Working with the spreadsheets in FCS Express isn't very difficult. And really what we're trying to do here is streamline that analysis to reporting while reducing copy and paste to other reporting and presentation tools, right? If you had to copy and paste every particular population where you wanted a MFI value converted to MESF, that would be kind of a big pain. But by integrating the channel calibration tool, reusing the templates, having the integrated spreadsheets, it all allows you to very easily get to publication quality results and graphics. And it's all done through a Microsoft-like interface, which makes it easy to use and learn. And of course, there's extensive online tutorials and training resources to help you get started with this stuff. But I would always say, if you have any questions, if you see something you like on this webinar and you want to implement it in your lab, write us an email at support@denovosoftware.com. Reach out to your representatives at Spherotech and ask them how to get in touch with us. We're always here to kind of help out. So with that, I hope everybody was able to take away a little information today about, what Spherotech has to offer in terms of calibration beads, in terms of alignment particles and alignment beads, what Novo Software has to kind of help, move those from just beads and raw data to final results. And we'll take a moment here to answer a few questions that kind of came through. But again, thank you for attending the webinar today, or thank you for viewing the recording afterwards. A big thank you to Michael Collis and the team at Spherotech for making this webinar possible. And again, we really appreciate your attendance today, and please let us know if you have any questions.